Without enzymes, vital chemical reactions would take millions of years to occur. With them, they happen in milliseconds. Enzymes are highly folded protein molecules, acting as biological catalysts to speed up these reactions. They're never consumed. They remain intact, ready to be used again. To understand how they achieve this, we need to look at the energy required to start a reaction. Every chemical process requires an initial push to get going. We call this activation energy. This diagram shows activation energy as a physical hill. Molecules need enough energy to get over that peak to complete a reaction. An enzyme physically lowers this energy barrier, reducing the initial energy required so the reaction can proceed rapidly. So enzymes do not create new chemical reactions out of nothing. They simply manipulate existing energy barriers, allowing vital processes to occur efficiently at normal body temperatures. You can usually identify an enzyme by its name, which typically ends with the suffix ase, like amylase or lipase. The first part of the name tells you exactly what molecule it targets. Enzymes are highly specific. A single enzyme will only ever work on one specific molecule. This specificity is driven by physical shape. The target molecule an enzyme works on is called the substrate. The enzyme itself features a precisely shaped cavern on its surface called the active site. When the substrate collides with the enzyme, the geometric contours of the active site wrap around the molecule with total precision. This creates a temporary, unified structure called an enzyme-substrate complex. Once bound, the enzyme facilitates the reaction, breaking the substrate apart or fusing it together. Here, the substrate is split into two new product molecules, which are released. The enzyme itself is left completely unchanged and immediately cycles back to find another substrate. The entire system relies on that precise 3D geometry. If the physical shape of the active site does not perfectly match the substrate, no complex forms, and the reaction will not happen. This chart tracks the rate of an enzyme-catalyzed reaction across changing temperatures. On the left, we map the overall speed. On the right, we see the molecules themselves. At low temperatures, kinetic energy is minimal. The enzymes and substrates move sluggishly, resulting in very few collisions. As the ambient temperature rises, thermal energy is converted into kinetic energy. The molecules move faster, the vectors grow, and they collide with the active sites more frequently, steadily driving up the overall rate of reaction. This acceleration continues until it hits an exact peak. This is the optimum temperature, the point of maximum efficiency. For human enzymes, this sits right around 37 degrees Celsius, perfectly aligned with our normal body temperature. But look at what happens immediately after that peak. The reaction rate plummets. Excessive heat introduces so much kinetic energy that it physically breaks the internal bonds holding the protein together. The active site warps, and the substrates simply bounce off. We call this structural collapse denaturation. It is the irreversible loss of the enzyme's 3D shape. Once an enzyme is denatured, it can never bind to its substrate again. Excessive heat permanently dismantles the molecular machinery required to make the reaction happen. Let's test that logic on a new variable. This chart maps the reaction rate against changing pH levels rather than temperature. Knowing that highly acidic or highly alkaline environments also break protein bonds and warp active sites, what should this curve look like? Take a moment to predict the shape. Because both high and low extremes of pH cause the enzyme to denature, the data forms a symmetrical bell curve. The enzyme only functions efficiently at its specific optimum pH in the center. Notice the critical difference in how extremes affect enzymes. Cold environments cause reversible inactivity. Warm the enzyme up, and it works again. But extreme heat or extreme pH causes permanent, irreversible denaturation. To master enzyme problems, always rely on these core principles. Enzymes lower activation energy to speed up reactions. They require precise 3D geometry to bind specifically to their substrates, and their efficiency is completely dictated by environmental conditions like temperature and pH.